going to need your foam dissecting tray, plastic knife, a magnifying glass, coating pins, flowering plants such as alstroemeria, or another name for this flower is Peruvian lily. So we're going to dissect the flower parts starting from the outside, and we're going to work our way in. So the very first part that you're going to dissect is the sepals of the flower. Be very careful as you're removing the sepals that you're only removing the sepals and not any other part of the plant. Now let's dissect the petals. With the same procedure, we're going to remove gently the petals. So this is what it will look like with your sepals and petals dissected and labeled. Notice the colors correspond with the colors on your coating sheet. Next, we're going to dissect the stamen, or remove the stamen. And remember, this is a monocotyledon, so how many stamens should there be? Right, there should be 6. Now some of them look a little different, but these are all the stamen here. So you're going to remove them from the bottom, careful not to remove any other parts except the stamen. Make sure that you do not dissect or take off this part yet. Use your magnifying glass to look at the differences between the stamen. Some are long and thick at the top, and some are kind of short and round at the top. And you notice if you touch those ones, you get a little bit of pollen on your hand. So looking back at your coating sheet, you'll notice that the parts of the stamen are coated with the yellow pins. The anther is coated with the yellow pin with the black dot, and the filament is coated with the solid yellow pin. Take a look at the stamen of some other flowers. Notice they're all just a little bit different. So what's left of your flower, still attached to the stem, is what's called the pistil. And the pistil is made up of the ovary, the style, and the stigma. You are going to make what's called a cross section of the ovary of the pistil so that you can see inside. So what you're going to do is you're going to anchor the pistil to your dissection board. Then you are going to carefully saw back and forth, or cut, with your knife. Careful not to cut this part of the plant off, the style of the plant. You want to keep that intact and whole. And that's where you can see the ovary. So looking back at the coating card, you'll notice that you'll need the orange pin, the red pin, and the red pin with the dot to label your pistil. So remember, you are going to record all your observations from this flower dissection into your science journal by creating and labeling a diagram of your own. If you have a flowering plant in your yard, ask your parents if you can dissect it at home, under their supervision, of course, and see if you can find all of the same flower parts that we did in this dissection on the flower that you got out of your own yard.